The overall goal of this invention is to study the effects of hypoxia or hyperoxia on the embryonic tissues of any aquatic organism of interest.
This method can help answer key questions in the field of developmental and cell biology, such as embryonic growth restriction, fetal hypoxia, high-altitude adaptation, obesity, and solid tumor development. The main advantages of this technique are simplicity, cost-effectiveness, and robustness. It allows induction and sustainment of hypoxia in vivo for long-time periods.
After collecting and raising xenopus and zebrafish embryos, prepare a mesh-bottom 24-well plate according to the text protocol. Attach plastic rods to the mesh-bottom 24-well plate and place it into the aquatic tank of choice. Then, using silicone tubing, attach a gas tank with the desired oxygen-nitrogen mixture or another gas mixture of choice to a distributor valve using an appropriate gas regulator. Next, connect the silicone tubing and the distributor valve to a ceramic disc diffuser inside the aquatic tank. Depending on the type of embryos used in the experiment, fill the hypoxic chamber tank with the appropriate embryo medium. Then, close the aquatic tank and use silicone grease to coat the lid and carefully seal it. Start diffusing the gas mixture through the ceramic disc diffuser. Allow the system to equilibrate for 10 to 15 minutes. Adjust the oxygen flow rate in the chamber medium to 0.0017 to 0.0019